And so I think Joey and me already answered quite a few of them, not all of them. I'm also not sure if everything is relevant exactly. So there's a few questions about Monte Carlo simulations at the beginning. So what would be a good amount of Monte Carlo simulations? And then also one question is, I think Eric also answered on that. And one question is, can you explain a bit more what's actually happening in Monte Carlo simulations? Do you think we should go into that, Eric?
I only read question 1 and 2. I think for question 1, there's a good answer, I think. I think since a few years, we now report the Monte Carlo confidence interval for the permutation p-value.
Okay.
And
do you want to explain what it is and how people get it?
Yeah, okay. So, a difference between Neyman-Pearson and the permutation approach is, on the practical side, is that in Neyman-Pearson you get an analytic p-value. That is, you can calculate the tail area probability using mathematical functions, and they are very precise, up to machine precision. That is not the case for a permutation p-value, because that is actually based on random sampling. So you randomly permute your data, and then for every random permutation, you calculate a test statistic, and that gives you a histogram of test statistics under the null hypothesis. And under that histogram, you then calculate your p-value. And you can imagine, if you have a small number of random permutations, that p-value is inaccurate. Fortunately, we also know exactly how inaccurate it is. And the inaccuracy of a Monte Carlo p-value is quantified by the Monte Carlo standard error. And if you have the Monte Carlo standard error, you can also calculate the Monte Carlo confidence interval. And that confidence interval, that will shrink to 0 if you increase the number of random draws from the permutation distribution. So this is actually only relevant in practice if you have a p-value which is very close to 0.05. So if you have a true p-value which is 0.049, you actually need a very small Monte Carlo confidence interval to evaluate whether it is less than 0.05 or not.
Do we get that in the stat output by default?
Yeah, I think so. The last time I looked at it, it was in there, yeah.
Yeah, I think so. If we still have one. So if you look at stat.
It's in one of the fields of the output. It should say something like Monte Carlo confidence.
I think CI range, I guess, or?
Yeah, CI range, that's it. CI is for confidence interval.
Yeah. Let me just add something here, because increasing the number of permutations is possible if you have a lot of observations, so if you have a lot of trials. But if you're doing an analysis at the subject level, then the number of subjects might actually be the limiting factor.
Yeah.
If you have one subject, you can't. If you have two conditions within one subject, you can only shuffle them in 2 different ways.
Yeah.
If you have 2 subjects, you can shuffle them in 4 different ways.
Yeah.
But it means that if you have 8 subjects, you can shuffle the 2 conditions within the subject in 56 ways, which is actually not a lot.
Precisely.
So I would say that at the group level, you need at least 10 participants.
Yeah.
So then you can do 1024 shuffles.
Yeah.
And then it starts becoming like interesting. Yeah. But I would say 10 is really the minimum, and preferably to have more participants.
Yeah.
For shuffling. So if you have so you could specify a number as the number of randomizations. You could also specify the string all. In the same way as you can, like CFG to channel, you could specify all. You can do the same thing with CFG non-randomization. And then it's going to make an exhaustive list of all possible permutations. And that is useful if you have like a reasonable number of subjects. I think it would work up to like 16 subjects or so, because at a certain point, the number of randomizations is just going to get too large, and then the computation time would get too long.
Yeah.
So that's also something to consider, like practical consideration.
Okay. Maybe we can also add that here. So
does it also mean that, sorry. Does it also mean that if we increase the number of conditions, we can increase the number of permutations?
Tibor, how would you increase the number of conditions?
I mean, obviously, during the statistical design, so before the acquisition, obviously. So if I have an experimental design which have more than 2 conditions, then I can do more permutations, right?
You can do that. I think from a point of view of obtaining an accurate p-value, this is a good thing. But I'm not such a big fan of statistical tests of independent variables with more than 2 levels, because they're very difficult to interpret. I don't like an f-test, actually. If you have, say, more than 2 levels, you have a significant f-test, then the next question is always, which levels are different?
That's true, yeah.
I like t-tests, and that's for non-statistical reasons. It's just because it's much more easy to compare 2 levels than to compare 3 or more.
How about if you come up with a contrast? So if you not just if you fit a model into it, and you say you are saying not an f-test but more than a t-test when you say you want to test this particular effect, let's say a linear effect over the 3 conditions.
Okay, if you have quantitative levels, that's a different ball game, because then you typically have some kind of a model, some kind of a model of how the quantitative levels relate to the measured variable, the biological variable. Then you enter in the field of also psychophysics, which they have models that differ with respect to the type of relation between the quantitative levels of your independent variable and the expected behavioral or neurobiological response.
Yes.
Yeah, sure. But for that, you wouldn't use actually an f-test. You would use a single degree of freedom test, and that test should be only sensitive to deviations from the expected quantitative relation. Okay.
Okay. Should we move on?
Sure.
So I saw we only have 6 questions total, I think.
Yeah.
The next one would be a bit more explanation on Monte Carlo in general. Would be nice for me personally, but that we can also skip it. So Joey has given a more abstract explanation, or like with this with a drawing balls from an urn, for example. I've given you a very short code how you could just implement it yourself, basically. I'm not sure if it's good. Maybe you can just have a look at that, or the person who asked this question can maybe ask what exactly he or she wanted to know.
Unmute button is hard to find. Yeah, what I exactly wanted to know, I'm not quite entirely sure. I am not a statistician in any way. I just have a lot of courses in it. So it's not intuitive for me. I need to learn it really from the ground up. And I do understand the answer, but I don't yet fully understand how Monte Carlo estimation works, because if I would have a vase and just grab random balls from it, would that already be a Monte Carlo simulation? Because that I have never learned as such. Or is it something different with something special in it? That I don't know. And if it's too difficult to explain it or too easy, then it's fine to skip it.
Well, I'm sure the Monte Carlo there's more than just randomization and permutation, and the script which is here is basically just a randomization and permutation. But I don't think that any permutation is a Monte Carlo. So the Monte Carlo is a bit more than that. But I'm also not expert enough for that.
Maybe Eric can comment on that.
Sure. Should I comment, Johannes?
Yep. Yeah, please.
Okay. My problem, Jefta, is that it's not completely clear to me what is unclear to you. So I could say so many different things, but I do not know which of these different answer options would help you.
I think that is my problem too, but the other way around. So I am not sure what to ask because I'm just not really well versed in math, and specifically this kind of math.
Are you asking for the concept or the algorithmic description?
I think rather the concept, but if I just can get it by Googling or by
that's I would say what Robert just posted.
Yeah, maybe
exactly.
we should just move on, and then we can ask. You can ask it later, maybe again, if you still have time left, or in a personal chat with Eric or me. The next question is very practical. It's about the pipeline and when you would compute the neighboring structure. My answer was basically just to so hopefully you're gonna have one final setup of channels that you want to include in all your NL lists. So you just compute it once, and I would just compute it directly before you do the testing. Yeah, so you can just compute it once, and then hopefully you keep it for all your analysis. It's a bit weird if you have different channels for different analysis. It's a bit hard to compare them. But yeah, that's just practically, yeah. You only have to do it once. Not separately for every test, but just do it once.
Yeah. So doing the clustering only makes sense if you have the same channels recorded in the two conditions. And if you have the same channels, then therefore also the neighborhood configuration for the two conditions is equal. Actually, the channels and the neighborhood configuration is shared between the two conditions. Otherwise, the whole procedure doesn't make sense.
Yeah, that's already the next question. So the next question, I was not really sure. The person asked, so if you have already separated your data for the conditions, do you apply FT prepare neighbors to the conditions separately? Or is this done on all data? So I think this should be, as Eric said, should be the same neighborhood configuration for all conditions.
And that actually holds for all kinds of for all type of comparisons that you would perform. Not only comparisons by means of cluster permutation statistics, but simple descriptive comparisons also. If you would compare condition A, where you only have recordings from the frontal electrodes, with condition B, where you only have recordings from the posterior electrodes, that doesn't make sense. If you want to compare the two conditions, you must have recordings from exactly the same channels, and only on those channels you can make a comparison. With respect to those channels, you can make a comparison. For non-overlapping channels, difference is not even defined.
Let me also add something here from a practical point of view, which has to do with FT prepare neighbors. Is that the specification of neighbors is related to where the electrodes are, and as such, if you have a specific electrode cap, then you already know what the neighbors are before even having started your recordings, or at least you can already specify what the neighbors are. So I've just, in the Zoom chat, I've just pasted a link to Fieldtrip website. If you look on the documentation, then you can find the template documentation, and there's also neighbor templates. And we have a bunch of neighbor templates for systems that are well characterized, such as the CTF 151, the CTF 275, but also for the 1010, the 1020, and the 10.5 system. But yes, thank you. And these are included in Fieldtrip. But quite often it's the case that people have their own electrode layouts. So that's why we often call FT prepare neighbors, because you have to think about what you consider to be neighbors. It's not something that you can leave to us, because we don't really know your electrode layout. So
can I add something to, oh, sorry.
Yeah, please go ahead.
Yeah, something to the origin of my question. It was mainly because I wanted to know what type of data input the function required. So I now understand that if you do a split per condition, you would actually get the same data structure as the all data variable. So that was the question.
So FT prepare neighbor takes the data structure input, but actually the only thing that we care about is in the data.grad, so the gradiometer definition, because the data.gradiometer definition, that is where the spatial information about the MEG channels is kept.
Yeah.
If we would do it on the EEG data, then often we have the electrodes in a separate structure because they're not stored in the same file, and then we would specify it differently. So it is something that you only have to do once for your whole experimental setup.
Yeah, and so I think this was also related to the question before. So where in the pipeline do you call the function? So actually you could call it already after defining your data variable, right? So right in the beginning after.
Yeah.
Okay. Yeah. Thank you.
It comes a bit back to the question that was there on Monday about different systems. So imagine you do a study where you have pilots, patients and controls, and you would use a different system for the patients than for the controls. That would become very difficult to make inferences about. So if you would do a statistical test on two data objects, but they have different settings, different channels, for example, it's not so trivial to compare those two then. Yeah. So ideally, this is the same for both conditions. Okay.
Can I just ask, so what happens, this is probably more relevant for EEG, what happens if you say you have one channel that you have to exclude in pre-processing because it's too noisy? Then what kind of, would you not be able to, would you have to exclude it for all other participants, for example, or?
Can I take this question? Actually, if you remember the data set that we've been working on so far, it also had two broken channels, which were here on the left parietal lobe, more or less. So here at the back, there were two channels missing. And the consequence of this is that channels on the left parietal area, they have fewer neighbors. So it's more difficult to compile evidence for there being a significant effect over that brain region. So as such, with the data that we've been using in the playground so far, it's also not totally biased. We would be more sensitive to finding effects on the right, more sensitive to effects in the right hemisphere than effects in the left hemisphere. Well, in this case, it's language experiments or auditory experiments. So it's about temporal channels, but that's something that you would have to consider, especially with EEG. You would have more often channels like at random locations missing in different subjects. So what I then would do is, if I would want to do channel level statistics, I would interpolate those channels so that you have an even coverage of all the channels in all your participants, rather than leaving them out.
Can you do the same also if you, for example, would have different systems across subjects?
You technically could do that, I guess. If you compute a spatial filter, for example, and you sample that and predefined locations, you could do that. But it's quite a strong assumption that your estimation of the spatial filter works well or is comparable across systems.
Yeah. Actually, this is perhaps relevant for those of you working with an electroneuromag machine. The electroneuromag machine has the max filter software, and the max filter software is using a rather sophisticated mathematical approach for filtering out external versus internal sources. But the max filter software can do more than that. It can also project the data onto a standard position of the head of the sensors relative to the head. And actually, that's where this question relates to. Like, can you compute the magnetic field at locations where you actually have not recorded it? And that's what max filter does. There's other approaches for that as well. We have in FieldTrip, we have FT MEG realign, which does this. In principle, you could do the same thing for EEG as well. But what I would say is important in such a case is that you're not introducing a bias. So if you're going to use MEG realign in FieldTrip or max filter in the Neuromag software, you would have to apply it consistently on all your subjects so that all your subjects are affected by the interpolation just as much as any other subject. So you should not interpolate all your patients towards your healthy controls. You should interpolate your patients and your controls to the average of the patients and the controls.
Sorry, I thought the interpolation was done within a subject for missing.
Yes, but I think Tibor's question was more like if you have different systems, and then if you have different systems, that's why I was thinking that it was like for different subjects.
Okay. Is question 3 answered, sort of, with a neighborhood and when to compute it? I hope so. So question 4 got even a thumb up. But Robert, did you just answer that one before 4 o'clock? Or what is the... I joined too late, so I wasn't really sure what you're talking about.
Yes, that was indeed something that we were discussing in the general room with some participants. I can quickly summarize what we've been discussing. So of course, this specifically relates to statistics. So how is statistics different for continuous data? And I think the important thing is that with continuous data, you don't have the experimental manipulation within time or within trials. So what people often have as the variable of interest is whether participants are patients or healthy controls, for example. Or some other thing which is like a long-term change from, for example, EEG before having a night of sleep and EEG after a night of sleep, and see whether memory consolidation happens, or before and after pharmaceutical intervention. So I think that the important thing is that you would always do a comparison of different recordings versus a comparison within a recording. And the unit of observation would very often be the subjects and not the trial. But other than that, the whole statistical approach still works. The only thing is if you have continuous data, then time is not a variable of interest. So usually we do FREC analysis with config method as MTM-FFT to compute power spectra, but then you can still use clustering on the power spectra. So you can look, for example, without having to pre-specify that you want to look at alpha or gamma, you can use clustering, and then you can also increase statistical sensitivity without pre-specifying the frequency range of interest. So as such, a lot of the similar techniques apply, but it's just the structure of the data is slightly different in continuous data.
Okay. More questions on that, or should we move on? If there are no more questions, then we come to question 5, which is how to produce a design matrix for within subjects design with more than 2 factors. And I think Joey has already given an example. So I hope everybody kind of learned during the session that the design matrix differs a bit for between units and within units tests. So you have to specify a different test statistic. And the design matrix also looks a bit different, namely such that the first row is the identity variable, I think. Yes.
Can we just quickly check whether we agree here whether it's factors or levels? I think it's the question and the answer are a little bit
we skipped about that
here.
That's true. So the factors levels, because this is levels, I think.
This is 3 levels within 1 factor.
And the first...
My question, it was if you had 3 factors with multiple levels.
Okay. I think Joey also had the solution for that recently. So if Joey is still there, maybe wants to explain it.
I think Rachel's question, if it actually pertains to 3 or more factors, that requires a totally different type of answer than a single factor with 3 or more levels. What are the factors in your study, Rachel?
So in the study, there's an object on a table. So one of the factors, whether the object is changed or not. There's what the type of object is. So there's 2 different types. And then there's a different question you asked at the end. There's 4 different questions. So there's 3 factors with between 2 and 4 levels.
3 factors. So there, I'm sorry, what is, can you give a list? What is factor 1, factor 2, factor 3?
So the first factor is exchange. The second factor is the item. And the third factor is the question type.
Okay, and are they fully crossed? That means either exchange, not exchange. Then there is object type.
Yeah, it's a full cross.
Okay, okay, and I guess this is within participants' experimental design. So every participant has participated in. So how many objects you had?
2.
So it's 8 conditions. 2 by 2 by 2.
2 by 2 by 4.
2 by 2 by 4. Okay, okay, okay. Good. Well. Well, the way you do this here is by well, the number of elements or number of columns in your design matrix is equal to 16 because you have 16 cells. Yeah, and then you have 16 cells and you have 3 independent variables. And the independent variables are called EVAR. And so for every cell, that is for every column, you have to specify what is the corresponding level of the first independent variable, which exchange or not. The second independent variable, I think it's object type, which is between 1 and 4. And the third one, I don't remember what's the third one. 2 levels.
The third one was question with 4 levels.
Our question was 4, so 2 objects, so sorry. 2 by 2 by 4, and question was 4, yeah. That's how you organize it. And so actually, the construction of the design matrix, that is not the biggest challenge for you. For you, having a 3 factorial design of your experiment, it's going to be a challenge. Which are the relevant effects for you? So definitely there will be some interaction effects in which you have an interest. Yeah? And to answer questions pertaining to these interaction effects using permutation-based inference, that will require you to calculate the appropriate difference scores. Every contrast corresponds to a particular difference score. Yeah? And for every participant, you have to calculate this difference score. And at the end, this will amount to a whole series of analysis, all involving the dependent samples t-test. So you should take a careful look to that that frequently answered question page on interaction effects. And that will help you. Actually, I think that page was written for a 2 factorial experiment, and in your case, it's 3 factorial. So you will have to generalize the content a bit towards your own application. But that contains all the logic. Yep.
I think Robert has made a suggestion in the code, also in the answer sheet now also. So you see an abstract solution, you see a more concrete solution for 2 by 2 by 2 design, how to set up the design matrix. I'm not sure, so Robert, can you maybe also explain, so what is the null hypothesis that you would be testing here?
The null hypothesis would still be a hypothesis of exchangeability over all the different conditions. As such, it's not a very informative.
So it's 3 main effects.
No, it would just be, it would be an omnibus effect.
Okay, so omnibus across 3 main effects, I see.
Yeah, yeah. So which is also one that's not really interesting. So this is how you technically specify it, but statistically, given the experimental design, this is not really one that we would often use.
Okay.
But if you would want to do it, this is how you would specify the design matrix, and then you would get a then you would figure out that the data is not exchangeable.
Yeah.
So what?
And for Rachel, say you have an interest in a second order interaction effect, that would imply for you that you first calculate difference scores for 2 different levels, and then you would just use a design matrix for a dependent samples t-test. And the input to your analysis are difference scores. Yeah. So what Robert has shown now is an analysis. It's an analysis that you could perform on the outcome for each of the 16 different cells of your design. And it's an omnibus test. Is there a difference between these 16 levels? Or you could also use it for a main effect. Is there a difference between, say, the 4 different questions? But if you want to go really into what I think are the research questions that are in your mind, you would have to do some pre-processing, which involves calculating difference scores or contrasts. And then you would compare contrasts for 2 conditions between each other using a dependent samples t-test. These 3 factorial designs are quite uncommon in neuroscience. And the documentation for these designs on field trip, it's not extensive, just because they're so rare.
Yeah, I think so. I would elaborate on Tibor a bit. So in behavioral analysis, you might now, because you have 3 factors, you might look at all main effects and all possible interactions, but it gets quite messy. So here it might be helpful to be even more hypothesis driven and really think about what kind of main effect do you expect, what kind of interaction effect do you expect. So don't run, what is it, 2 by 2 by 4 and then factorial. It's a lot of tests, I think. Yeah, so it's almost 20, I think. So maybe not do that, but really focus on what you're interested in. Yeah.
I mean, in fMRI, we have such designs. Well, probably not a 2 by 2 by 4, but by myself, I have a 2 by 2 and a 2 by 2 by 2, for example.
Yeah.
So then you can look for how you can convert them into this kind of differentials going across the levels.
Okay. Is this question answered, sort of? Otherwise, maybe DM Eric or me, basically, and then, or Joey. Yeah. I see. Yeah. Okay. I see one final question, which I also haven't read yet, I have to say. So, if you have an experiment with the same number of units of observations between 2 conditions, would it be okay to perform a dependent t-test, even if the conditions are not actually dependent on each other? Or would it be recommended to run an independent t-test? E.g., instead of before versus after conditions, you just compare 2 different classes of stimuli. Yeah, this is an interesting question because so with the dependent variables or like dependent samples t-tests are quite common, and often it's just within a certain subject. So the most important part here, I think, is that the question is, does it make sense to compute differences? So if you have one subject,
the answer is simple, Johannes. It doesn't make sense.
No, but if you can still assign couples,
how would you? In a sensible way.
Yeah, whatever. If you have 2 subjects, but they belong together because they're married, for example.
Okay, but then the unit of observation is not participant, but it's couple. That's perfectly okay. Or twin.
Or twin, yeah.
In twin studies, you have this, but then you just change the definition of your unit of observation. Yeah.
Okay. Yeah.
So everything starts with the proper definition of your terms. Yeah? So you have to specify what is your unit of observation. It doesn't have to be a single biological organism, but you have to define it properly. Only after having defined your unit of observation, that's when the procedures come into play. And then the procedures, they have to obey your prior definition of a unit of observation. You with me?
Sorry.
Yeah. Is this sort of clear? So the thing is, so you must imagine, so if you have a dependent sample t-test, you have 2 columns next to each other, and the rows are meaningful. The row is 1 subject or 1 couple or 1 twin couple or something, yeah? But if it's sort of, if you have 2 independent columns, like one with your patients, one with your controls, and it's totally arbitrary how you would arrange them, the rows are totally arbitrary, then it doesn't make sense to compute a dependent samples t-test, I think.
Yeah, that's correct, Johannes.
Okay.
Just to say, I like the example of that we have that we can also have like gender and age matched samples. So it's any type of matching, and then that will be the unit of observation. It's a good extension.
It could be, yeah.
I asked this question because when I compare, for example, two conditions, the unit of observations were subjects, and I have the subjects passing through the two conditions. But I have, all the subjects passing through for the two conditions, I have the same number of units of observation for the two conditions. So if I do this task in EEG lab, the default is the EEG lab runs a dependent T task. But then I was just wondering if those two conditions are not like pre and post, would it be a dependency task actually, or an independency task? That's why I was like struggling with this.
But what are your conditions? Can you say that? I still don't get it.
So like I have, for example, fair versus unfair condition, fair versus unfair situation. And then I want to compare all the subjects pass through fair versus unfair situations.
Okay. So you have two data points per subject.
Yeah, let's say, yeah.
Okay,
yeah.
If each subject does each condition, then you can commute within subjects, then you would have a dependent samples test.
Okay.
Do you have a full randomization? So is the order of varies randomly before across participants?
Okay.
Because if all the participants has, for example, unfair first and fair later, then you will not be able to separate the temporal effect, time effect, from the actual condition effect.
Mm-hmm.
So I'm not sure if your question is related to that.
No, it's not exactly related to the order. It was what you're saying.
So order was randomized across participants.
It was, yeah.
Okay.
Yeah.
Okay. I think we've reached the end of this document, but are there more questions? Because we still have some time, I think like 10 minutes. Can I also stop sharing my screen?
Perhaps also practical questions that had to do with the hands-on session. So people might have like ran into problems executing the hands-on session, or have questions about like how to visualize these type of things, or again on the neighbors, which I know is like a frequently asked question.
I was just wondering about if you can just clarify again so I get the IVAR variable. I was just reading on the online, and the UVAR, and there's the WVAR. Can you just clarify how they help you set up the permutation? Basically, it's online.
Shall I try to do a graphical explanation just using a piece of paper?
Excellent. Yeah.
So, the way that we organize the data is like this. So this is the data, and what we do is we all the observations that we have within, so all the data points that we have within one observation, we represent it in a column. So this can be channels times frequencies, or this can be voxels if we have a source reconstruction. And then these will be either the different subjects or the different trials. So what we want to do is we have a hypothesis of exchangeability. So what we want to do is we want to randomly shuffle columns here. And then what we subsequently do is, if we have shuffled the columns, we compute some function of this. So we compute a function on this matrix, which for every channel time frequency point over every voxel computes a massive univariate statistical value. For example, an independent samples t-test. Okay, so this matrix is very large. So what we rather than shuffling this matrix, which would actually require a lot of like moving data around in memory, we're using the design matrix. So what we have here is that here we specify here we specify in the different rows of this matrix, we specify for each of the columns to which of the subjects or which of the units of observation it belongs, or to which of the independent variables it belongs. And then within, rather than swapping the columns of the data matrix, we swap the columns of the design matrix. And it's the UVAR, the IVAR, and the WVAR, and the CVAR that put restrictions on how the columns of the design matrix are being swapped. So what happens is that, for example, we would say, well, oops, I'm not drawing on my table here. So rather than swapping this column with this, what we're doing is we're swapping this column of the very small design matrix with this column of the design matrix. And then subsequently, so this function that we compute takes the design matrix and then does its computations. So that means that we don't have to move the data around, but rather we move the design around, and then we can still compute our massive univariate statistic under the hypothesis that these can be exchanged. So the private, so field trip / private / randomized design. So that's the function that does the shuffling. It basically looks at how you've specified IVAR, UVAR, and CVAR, and WVAR, and it puts on restrictions on how the columns are being shuffled. Actually, if you look in more detail in that function, you will also see that it does not only do randomization tests or permutation tests as we normally refer to them, because we're not doing random randomization, we're only permuting, so we keep the number of elements the same. But it can also do jackknifing and bootstrapping. So it does implement a few different resampling strategies, but the one that we tend to use, because that's the one for which we have mathematical proof that it results in the correctly interpretable p-value, is the permutation test. But if you would want, you could also use a bootstrap test to compute p-values, but those p-values are a little bit less well-founded. Does that answer the question?
I'll share this link as well, for which I just found.
Okay, any more questions or practical questions on the hands-on today? So some people had, I think, problems with the highlighting of significant clusters. That was sometimes the case because there were no significant clusters, which you could just check by looking at what is the content of your pos or pos-int vector, I think. I think the tutorial was a bit out of date or something. I think they changed something in between. But
can I just chime in here?
Yeah.
So I am just right now updating the figures in the tutorial, because there was quite some confusion because of the fact that the figures in the tutorial were outdated, using older version of field trip and old column maps and stuff. So that will become online in a few minutes, I think. And one thing to beware, so not meaning to be nasty to Johannes, but strictly speaking, it's not allowed to talk about a significant cluster. We do a test where we either decide that we reject our null hypothesis, so we decide our test is significant, and that is based on clusters. But
very important. Yeah.
He said it on purpose just to allow me to make this remark. So thanks, Johannes, for that.
Very good. Shall we then wrap up the Q&A session for the statistics hands-on? I realize it has again been quite a long day. We started on Monday morning with topics that were like reasonably okay for most of you, just like pre-processing, how are the different acquisition systems. But now we've actually ramped up quite a bit in terms of the complexity of the analysis methods that we've been using. So first, like event-related potentials, that's a simple one. But then frequency and then time frequency, then source reconstruction, beamforming. Now we've added statistics to the whole thing, and there's still one more thing to come in terms of like techniques that we want to explain to you. That's the connectivity analysis, and that's what we're going to do tomorrow morning. So we're going to have a connectivity lecture, pre-recorded, but Jan-Matthijs is presenting it. And then after that, we're going to do a connectivity hands-on. And I think, given like the research question I've read from you, that's also going to be relevant for quite some of you. So of course, tomorrow afternoon, we're also going to move on to the playground session. And Jan-Matthijs has sent around a questionnaire for the playground. I have not checked, I must say, I have not checked yet. So that's in the, yes, that's in the general channel on Slack. Actually, let me stop recording. It's not relevant.